Recording in progress.
Utaka vitamin D. Haya. Vitamin D.
Your microphone, please. Now, how do we get to know where this is? Sometimes we have bleeding in the brain. Certain areas of the brain will lack blood. So when they lack blood because where there are clots or that blocks blood supply to that area, then we see certain behavioral changes. And those behavioral changes, therefore, we know that this area must be associated with this particular function. Some of them are, for example, if you have primary motor area affected, we know the motor area control our motor activity. It is the source of our corticospinal tract. And the corticospinal tract synapse with the neurons in the anterior horn of the spinal cord, the motor horn of the spinal cord, that was derived from the basal plate that supplies the skeletal muscles. So if our primary motor area is affected, then definitely our muscles will not be able to work. We have paralysis. But we noted that within the medulla, there is decussation. The fibers from the right side cross to the left side, and the fibers from the left side cross to the right side. So that we refer to as the motor decussation at the pyramid area, close the pyramid area, in the medulla oblongata. And therefore, if our right side of the brain is affected, then the left side of our body will be paralyzed, and vice versa. Primary sensory areas. This discrimination, sensory discrimination, tactile discrimination, agnosia, astereognosis, that's being able to determine things in 3 dimensions. If you remember, we talked about spinothalamic tract, for example, going to the brain. We talked about the lemniscal system, and the lemniscal system, if you remember, there was a crossing over. There was a crossing over at what we are calling sensory decussation. Within the medulla, after the gracilis, nucleus gracilis, and nucleus cuneatus. So again, there will be contralateral loss of sensation, depending on which area is affected. For the spinothalamic areas, also we have general sensation. So again, this one here, you might lose your touch, but you might be able to maintain the discriminative touch, but not the crude touch. That means that the lemniscal system has been affected, but the spinothalamic system, if you are able to discriminate between two modalities, the density, gradient of touch, for example, fine touch, then it means the dorsal column is normal. It means our lemniscal system is intact, but you have lost the crude touch. That means that spinothalamic pathway has been affected. So we need to be able to understand that in terms of both ascending and descending fibers. The Wernicke area, we said, is sensory speech area, linguistic interpretation. You see a page, you see a picture, can you interpret that in terms of language? So to be able to do that, the information initially, you hear some sounds, can you interpret that? So that information of being able to distinguish is regulated within the Wernicke area. So if the middle cerebral cortex, middle cerebral artery is affected, which supplies this area, then there is that loss of linguistic interpretation. And remember, there is also what you call brain dominance. The left brain is dominant for language. If the left part of the brain is affected, then you have more loss of language interpretation than if the right was affected. The primary visual cortex, that is area 17, if that is affected, and that is from the posterior cerebral artery supplying that area, then you have loss of vision. You cannot be able to see if the primary is affected. But if the association areas are affected, then there is loss of visual recognition. You can see, but you cannot be able to interpret what you see. And then the other areas, you remember the medial geniculate body and so on, auditory areas, respiratory areas, which you talked about. So all these are important in terms of being able to understand why certain functions of the brain are actually affected. The piriform lobe is visual areas. We'll talk about vision, and we'll note that vision, the pathway for vision also goes through the temporal lobe. And within that temporal lobe, it is the piriform lobe. If there's a damage to that area, you might also have some visual problems. But temporal lobe is very important in memory, sometimes behavior, because within this area, we have the limbic system. This is a location for the limbic system. I think we have discussed that. Now, when we looked at the spinal cord, we talked about types of neurons. We talked about interneurons connecting the dorsal to the ventral. We talked about commissural fibers connecting one part to the next opposite. And we talked about association, projection. That is endogenous type of neurons that actually are interneurons. Within the cerebral cortex, we also have fibers. Remember, the fibers from the spinal cord, from the midbrain, from the brainstem, have to go all the way to the cerebral cortex. So this white matter area here represents myelinated axons. And the outer part represents the gray matter, the cell bodies. So this is the gray matter. And from this, you can even see the thickness of the various parts of the gray matter. Then internally, we also have some cell bodies, aggregates of cell bodies. These aggregates of cell bodies, in the case of the cerebral cortex, we refer to as the basal ganglia. So the basal ganglia, made up of the caudate, and the caudate has got a head, the head of the caudate has got a tail of the caudate. So it's head and tail. And then we have these other ones here, which, if you look at it, it's like a lens. So this is what you call lendiform nucleus. Lendiform. And this lendiform comprises of the putamen and globus pallidus. Putamen and globus pallidus. And externally we have this small area, the claustrum. In between this caudate and the lendiform, we have this white matter. This white matter is what we refer to as the internal capsule. The internal capsule. The internal capsule carries a lot of fibers of the cerebral cortex, various parts of the cerebral cortex. Our visual system passes through the internal capsule, the axons. Auditory radiation pass through the internal capsule, and very many others. So it's very important. It's a very important area. And that's why it's myelinated axons. The fibers within the cerebral cortex could be classified into two. We have into three, sorry. We have the commissural fibers. Remember, commissural fibers, when you looked at the spinal cord, are those which cross from the right to the left. So commissural fibers in the cerebral cortex are those which cross from the right hemisphere to the left hemisphere and vice versa. These commissural fibers pass, in the case of the cerebral cortex, form what you call corpus callosum. Corpus callosum. Remember, we talked last time about an anomaly where we have an absence of corpus callosum. But corpus callosum connects the right and the left parts of the hemispheres. And this is very important sometimes in sort of comparing the information so that you can take proper action. So those are commissural fibers. The other fibers are projection fibers. These projection fibers could be originating from the ganglia, for example, the basal ganglia, nitrostriate fibers, for example, from the substantia nigra. So these fibers project to the cortical, the gray matter on the cortical part. So those we refer to as projection fibers. Then we have those fibers which connect the various gyri. For example, this is a gyrus, and this is another gyrus. So we can have a fiber from a primary visual area, primary visual, connecting to visual association area. So that axon goes on to connect to the other one. So that's what we refer to as association fibers. And these are very important fibers which we'll be looking at later on in the cerebral cortex. At this stage, I'd like us also to look at this area here. You can notice this area is very thin, and this part contains a very thin rim of white matter and also gray matter. You can see a lot of gray matter here. So this part is what we call the Hammond's horn. Hammond's horn, like a horn of a ram. That's what it means. And this one here is part of the limbic system, part of the primitive cortex, what we were calling before the archicortex. If you remember when we were classifying. I'd like us also to look at this part here. This is the middle cerebral artery, but I'd like us, this part here is what we are calling the insula. Insula. So during the development and also that hemispheric rotation, this is an older part of the brain which had developed, but as the brain continued developing with that hemispheric rotation, this part comes to be buried. So you don't see it from the outside. That's what we are calling the insula. These are the spaces, are the ventricles, which we talked about. This part also is part of the lateral ventricle here. So the white matter fibers, the types are association fibers, commissural fibers, and projection fibers. I think that has been clarified. So we'll be talking about association fibers as we talk about cerebral cortex. We'll be talking about commissural fibers, and that's why I'm mentioning it at this particular time. And then projection fibers. Now the basal ganglia are groups of cell bodies buried amongst white matter, the internal part and the white matter. And this basal ganglia, remember when we were talking about it, they originated from both the ala and basal plates. So they moved, the axons, the cells moved. As the first group of cells that moved out, they formed what we are calling the mantle zone, in this case the intermediate zone. And then the second migration of the cells that form the cortical place bypass, so that those cell bodies differentiated to form the basal ganglia. I've just talked about them, and that is the basal ganglia that we're talking about here, the putamen, the caudate, the lentiform, which is globus pallidus, putamen, then claustrum. Can you mute your microphone, please? The allocortex. Remember, we classified the cortex into a neocortex and allocortex. So the allocortex we had classified again into the paleocortex. Sorry, we classified as archicortex and paleocortex. So the archicortex, the part I've shown you here, this is part of the archicortex, what we refer to as hippocampal formation. Hippocampal formation. Also some cingulate gyrus and area subcallosa. Area subcallosa is this part. So we're talking about this region here, part of the piriform, part of the temporal lobe. Area subcallosa, and then we have cingulate gyrus, the hippocampus is the important one. This is what we're calling the allocortex. The paleocortex will be the olfactory bulb. Olfactory bulb, the piriform lobe, and then part of the amygdala constitutes what you call the paleocortex, the ungus and the amygdala, paleocortex. These two constitutes a three-layered cortex. The archicortex constitutes a three-layered cortex, unlike what we see in the cerebral cortex. It's a more primitive cortex that developed very early, and that was mainly for survival. So this is the limbic system. For the histological part of it, we think about it as the molecular layer. Outermost is molecular layer comprising of mainly glial cells and synaptic conducts. So it's more of an association. This is where we have a lot of synapses. Then the granular layer, which is a receptory area, or pyramidal layer, either granular or pyramidal layer. Now, in the case of the hippocampus, here, we have a molecular layer, then we have a pyramidal layer. And the pyramidal layer consists of large pyramidal cells. These pyramidal cells are sources of cells. The axons from these pass very close to the ventricle, forming what we refer to as the alveus. And those ones connect now to what we refer to as the fornix. Connects to the fornix so that we can now have that information going to the rest of the cerebral cortex. In the case of the dentate gyrus, instead of pyramidal cells, we have granular layer. And then after that layer, molecular layer, pyramidal layer, then we have a sort of mixed area of neurons. This is our hippocampal formation. This is the hippocampal gyrus here. And this part here is our dentate gyrus. When you look at this dentate gyrus, it gets its name from the tooth. It looks like a tooth. That's why it's called dentate gyrus. And these closely packed cells here in the dentate gyrus are granule cells, large stellate cells, a lot of processes. These are granule cells. These other cells here are the pyramidal cells. So this is the hippocampus, and the hippocampus consists of pyramidal cells. These pyramidal cells continue on, show you again in another slide, will continue on. It will continue on, and you can see here, there's a change. There's a change. This we have said is the dentate gyrus. This one here is hippocampal gyrus. So this part here is a molecular layer. This one here is the pyramidal layer. Pyramidal layer. That pyramidal layer continues on, and this is an area of mixed neurons, therefore polymorphic layer. For the hippocampus, this continue on, molecular layer, pyramidal layer, and then the polymorphic layer. But in this position, a place they call parahippocampus or subebiculum. You can see that those pyramidal cells now spread out, and we have extra neurons here. So this is a transition from the 3 layers to the 6-layered cortex, and this now becomes parahippocampus. Parahippocampus. This is part of the limbic system. Just another picture of the cell. Remember the dentate gyri. Dentate gyrus containing granule cells. Just like what we are seeing. These ones here are granule cells. These ones here are pyramidal cells. So the dentate gyrus will also have a molecular layer, it will have a granule layer, and then a polymorphic layer. So it is 3 layers, but instead of pyramidal like in the hippocampus, it has granule cells, both acting in the same manner. These cells, the pyramidal cells here, are the effectory. They are the effector. So this output from the hippocampus. Now for the paleocortex is a relatively later development. So we had our archicortex developing, then paleocortex, phylogenetically. So a paleocortex comprises of regions of 3 layers up to 5 layers. So sometimes part of what is called parahippocampus could be considered as a paleocortex, a transition, a graduation from a more primitive to a less primitive type of cortex. Our olfactory bulb, comprising of almost 6 layers. The olfactory bulb, olfactory tubercle, piriform cortex, which is part of the olfactory system, and then the amygdala. Amygdala is mainly responsible for aggressive behavior, emotions, part of the limbic system. Now this comprises of 3-layered cortex, like what we have just seen, a molecular layer, pyramidal layer, and then a polymorphic layer, up to 5 layers, like what we have in the olfactory part of the olfactory. So depending on the areas, we have this graduation of the number of neurons to now a more advanced type of new cortex that we refer to as the neocortex. So let's look at that neocortex. What is that structure of the neocortex? Our neocortex, remember, developed from the cortical plate. Cortical plate. And that cortical plate was formed by the inside-out layering. in terms of migration of the neurons. In our motific cortex, we have 6-layered cortex. 6-layered cortex, I've just used this as an abbreviation. MGP squared poly area. So these are the layers. The first layer we have is a molecular layer. Just like what we saw in the case of the cortex, we have a molecular layer, and this molecular layer is mainly made up of glial cells. Glial cells, these ones are astrocytes, oligodendrocytes, microglia, and then we have horizontal cells of Kagel. Horizontal cells of Kagel. These horizontal cells act as interneurons. So in this molecular layer, we have a lot of synapses, and therefore this is an association area. That the A stands for association. M is molecular. Then we have the outer granular layer, G. What I put there as G is outer granular layer. And this one here consists of small pyramidal cells. Small to medium-sized pyramidal cells. Small to medium-sized pyramidal cells, and also Golgi type 2 type of neurons. This is where we have, it's a receptor area. Some of the fibers coming from the various parts of the body will synapse in this area. This is where we have the reception. Some of the fibers synapse to this area. That's why we call it a receptor area. Small r. I'll explain to you why that is so. Then the outer pyramidal. The outer pyramidal consists of medium-sized pyramidal cells. Medium-sized pyramidal cells, and also some Golgi type 3 cells. Golgi type 3 cells. And some other cells we call Martinotti cells. These are actually neurons that have specific names based on who described it. So somebody called Martinotti described those ones. Now this one here is both association area, and commissure. So commissure means they cross the midline, fibers going to the opposite side of the cerebral cortex, to the cerebral hemispheres, cross, and they originate from these small pyramidal cells. So remember that association. Now, what are these association fibers? The association fibers here are going to the very other parts of the brain, like what we had described before, as association fibers connecting various gyri, various parts of the gyri, but from the same side of the brain. And then there are those who cross the midline. That's why we call them commissural fibers. That is the A. Then we have the inner granular. The inner granular layer consists of stellate cells, stellate neurons, and also some small pyramidal cells, but mainly stellate cells, more closely packed stellate cells, and some pyramidal cells. So this area of the inner granular is the main receptory area. That is this R, main receptory area. This is where thalamocortical fibers terminate. From the thalamus to the cortex. Remember, all sensory information pass through the thalamus, and then from the thalamus, we now have a third order neuron, sometimes a third order neuron, which now project to the cortex. So this is where we have the termination of thalamocortical fibers, synapsing with these granule cells. Those granule cells are going to synapse with the pyramidal cells, and so on. And then the inner pyramidal, also called ganglionic layer. The inner pyramidal layer consists of in some areas, very large pyramidal cells. Very large pyramidal cells. Those large pyramidal cells we refer as base cells. And this one here is in Brodmann 4. So this is the thickest part of our cortex. Very large base cells. That's Brodmann area 4, which is the primary motor area. Primary motor area. And then the dendrites from this project to the molecular layer. They project to the molecular layer. Dendrites from this project to the molecular layer. And as they project to the molecular layer, notice they will be synapsing with others within the molecular layer. And then the axons from this form association fibers, the various parts of the cerebral cortex. They also form commissural fibers, projecting from one hemisphere to the opposite, and then projection fibers. Now, this is the main effectory area. So the output from the cerebral cortex is from the inner pyramidal layer. And this now is the origin of the corticospinal tract, corticobulbar tract, corticopontine tracts, which we talked about. Corticospinal, corticobulbar, corticospinal, corticobulbar, and corticopontine. We talked about when we were talking about the basal part of the bones. You can see their origin is from this inner pyramidal. Those fibers now, as corticospinal, they descend on to form the pyramid in the medulla, and then synapse, like I've said, to the basal part of the spinal cord, the basal horn of the spinal cord, the motor horn of the spinal cord. And then we have a polymorphic layer. Now, polymorphic layer contains smaller neurons. Smaller neurons, they are mixed type of neurons. Some are pyramidal, some of them are oval, some of them are pyramidal. That means they are rounded sort of neurons. And these ones are the main source of corticothalamic tract. Corticothalamic tract. Remember, there was thalamocortical tract, giving the information to the thalamus. From the thalamus, the cerebral cortex. Now we have the information from the cerebral cortex to the thalamus, a feedback sort of mechanism. So this is what we have as the structure of our homotypic isocortex. This is a new cortex, neocortex, as opposed to the allocortex. Now, for the basal ganglia, I had mentioned the basal ganglia, but let's just summarize what we have for the basal ganglia. They initiate motor activity stemming from the knowledge of movement, what we have already stored as information. That is the putamen and globus pallidus. So we have putamen, and then globus pallidus, remember? And then what was originated from the diencephalon, the coded nucleus from the diencephalon, and the claustrum. All these are aggregates of cell groups. Among these, although it's not quite referred to as basal ganglia, we have substantia nigra, which we talked about in the mesencephalon and the red nucleus. So the basal ganglia, together with the substantia nigra and the red nucleus, constitute what we refer to as extrapyramidal motor pathway. Extrapyramidal motor pathway. They regulate motor activity, but not through corticospinal tract. They fine-tune that motor activity. So you need to note that, and you need to note also that should there be any damage to this area, then that motor activity will be lost. You'll have lack of fine control of motor activity. From here, let's look at a structure associated with this, in terms of motor function, and how these basal ganglia help in fine-tuning motor activity within the cerebellum. So cerebellum is what they call some little brain. Cerebellum is a little brain. Although it's called little brain, cerebellum constitutes 50% of the total neurons of the brain. 50%. Why 50%? Most of our activity of the body is associated with movement. Most of our activity, think of it. Because it's associated with movement, we have to fine-tune that movement. We have evolved to be able to do some intricate type of movement. Be it walking, be it moving your tongue, and so on. So that is what we have as main function of cerebellum, mainly regulation of movement. Although it's very small, that's why it's called little brain. But you can see 50% of the neurons are within the cerebellum. Now, what you see from the outside of the cerebellum is only 15%. 15%. The rest is hidden inside within what these leaf-like structures that we refer to as the folia. But what are these movements of the cerebellum that we are noting? We say it's mainly coordinating muscle action. Coordination of muscle action by monitoring other brain centers. How does it monitor other brain centers? If you remember, we have brain centers where we have the nuclei in the brain stem. For example, the vestibular nucleus, which is very important in balance. We have within the cerebral cortex areas that are important in muscle tone, regulation of muscle tone. So information is going to the cerebellum from various parts of the brain, both ascending and descending fibers. The spinocerebellar fibers come from the spinal cord, the brain. We have corticocerebellar fibers, but these corticocerebellar fibers actually do not go direct to the cerebellum. They synapse at the pons, where we have the corticopontine fibers. They synapse with the fibers, both mossy fibers and climbing fibers, which then go to the cerebellum. So by monitoring other brain activities, they can then modulate muscle activity. That modulation of muscle activity will involve both muscle tone. Our muscles are all the time in tone. Both extensors and flexors are in a muscle tone, so that neither of them is completely relaxed. Or very fine movement, and therefore being able to balance and posture. What is posture? Posture is the attitude of the body in space. Whether you are seated, whether you are walking, that's part of your posture, your attitude in space. So as you move, as you sit, the cerebellum is all the time monitoring the tension in the muscles, tension in the joints, joint capsule from other centers of the brain. And then it's passing the information. Within the muscles, if you remember when we were talking about muscle tissue, we talked about muscle spindle apparatus that detect the stretch in the muscle and can initiate stretch in the muscle, both types of intrafusal fibers. We talked about Golgi tendon apparatus, detecting the tension within the tendon. so that it can either relax the muscle if there's too much pressure. And in doing that, we are able to have a proper muscle tone and therefore be able to maintain our posture. Whether we are walking or sitting. Static posture, where we are not moving at all, or when we are moving, a kinetic type of posture. So it's mainly for coordination of voluntary movement. Now, voluntary movement, remember, is associated with skeletal muscle. Voluntary movement. It's also important in motor learning and programming. When a child is born, they crawl, they do the first step, they attempt to walk, they fall down, they attempt to walk, they fall down for the first time. They are learning how to walk. That information is being stored until eventually when that child is able to walk properly, then that information has already been stored. You don't need to think of that type of walking. It's already programmed. The way you walk, it's already programmed. When you move your left foot forward, your left limb forward, you move your right forward in a matching situation. And like what a giraffe does. So that's already a programmed learning which has been stored, it's memory, and therefore it is retrieved whenever you need. Cognitive function, language. Language in terms of movement. Now, the cerebellum, remember, originates from the rhombic lip. Rhombic lip, that's neocerebellum. And also, part of it, they say, originates from the mesencephalum. That's a primitive part, especially the vermis. That is the paliocerebellum. Paliocerebellum is a more primitive type of cerebellum. That's the vermis that originates from the mesencephalum. The rest of it is from the rhombic lip, specifically the metencephalum. Well, you know the location of the cerebellum is the posterior fossa, cranial fossa, posterior cranial fossa, closely associated with the occipital lobe, separated from the cerebral cortex, the occipital lobe, by the falx cerebri, and attached to the pons by pendangos, both the middle, the inferior and superior pendangle. It's covered by meninges, falx cerebri, tendorium cerebelli, separating it from this occipital lobe, and falx cerebri separates the two cerebellar hemispheres. The two cerebellar hemispheres. We have a longitudinal fissure, and in that longitudinal fissure, we have the falx cerebelli. So we had looked at those pendangles before, inferior cerebellar pendangle connecting to the medulla, from the olivar nucleus, olivocerebellar tract, spinal cord, spinocerebellar tract, the cuneate nucleus, connected to the cuneate nucleus, trigeminal nucleus, vestibular nucleus. So you can see that connection, vestibulocerebellar, so that you can be able to balance. The middle cerebellar pendangle, which is pontocerebellar tract, and also origin from corticopontocerebellar tract. Corticopontocerebellar tract, which I just talked about, from the cortex to the cerebellum. We have synapsing in the pontine nucleus, then the mossy fibers and climbing fibers then transmit the information to the cerebellum. Then from the tectum, remember we have from the superior colliculus, we have to be able to move our necks, our head, and therefore the body must be in certain posture. That's why we have tectocerebellar from the tectum, from superior colliculus. Superior cerebellar pendulum group is mainly to the midbrain. Dendrobuthalamic, that's the dentate gyrus, the red nucleus, the thalamus. Tectocerebellar from the tectum, remember, part of the inferior colliculus. And the inferior colliculus, we said, are responsible for auditory reflex activity. So when you look closely at the cerebellum, this is our cerebellum, the little brain, you can notice that we have a lot of these leaf-like structures that we refer to as the folia, cerebellar folia. These leaf-like structures are separated by fissures. Large areas are separated by fissures. We have a horizontal fissure. We have a longitudinal fissure where we have the falx cerebri that now lies between the cerebellar hemispheres. Internally, right in the core of the cerebellum, is the first migration of cells from the matrix zone during the development of the cerebellum. These ones formed the mantle zone. And that mantle zone, what remains as the mantle zone, are actually the nuclei of the cerebellum. There are several nuclei of the cerebellum, the dentate nucleus, the emboliform nucleus, and so on. Those ones are at the core, are in the white matter. Then the second wave of neurons bypass that mantle zone and form the cortical plate, and the cortical plate now develop into cerebellar cortex, the outer gray matter that we refer to as the cerebellar cortex. So we have the vermis and the fastigial nucleus. For a median part, we have the globus nucleus and the emboliform nucleus. All these nuclei, like I've said, are very important relay stations for the fibers going to the cerebellum and the fibers coming from the cerebellum. The vermis, which you said is a primitive part, is mainly for movement of the trunk, limbs, and hands. Then the intermediate part is mainly discrete unilateral movements, which we talked about. The lateral zone is coordinating muscle movements. Muscle movements through the red nucleus, the thalamus, cerebral cortex. Now, this cerebral cortex, part of it, is also through the basal ganglia in the cerebral cortex. Like we talked about cerebral cortex, cerebellum is also classified into very primitive part of the cerebellum and the neocerebellum. We have what we call archicerebellum, mainly for vestibular system, and then paleocerebellum from the joint receptors, exteroreceptors, pressure, for example, from the skin and so on, from the head and body muscle tone. Also muscle tone, this regulates now the muscle tone, paleocerebellum. Then you have the neocerebellum, which is the cerebellar cortex, the new part. And this from the inferior olivary nucleus, that is very important in basic movement. That movement where you have fine-tuning the movement. Physiologically, when you look at the cerebellum, we have the cerebellar cortex, the whole of this is cerebellar cortex, and then the medulla. The medulla consists of myelinated axons, and within that medulla we also have oligodendrocytes responsible for myelination. We have astrocytes responsible for blood-brain barrier. We have microglia, which are responsible for clearing any debris, dead cells, and proteins. So that is the medulla part of the cerebellum. Then the cortex, cerebellar cortex, is made up of three layers. We have this outermost layer, which we refer to as the molecular layer. Molecular layer. Then we have a single layer of cells here, single layer of Purkinje cells. We refer to this as ganglionic layer. Ganglionic layer. And then we have this layer where we have stellate cells. This is the granular layer. It looks like grains. That's why it's called granular layer. So all these layers of the cerebellum, you can have a functional column. And that functional column is around a pyramidal cell. Because the pyramidal cell is the only output from the cerebellum. Only output from the cerebellum is the pyramidal cell. The other cells within the granular layer and also the molecular layer are either inhibitory or excitatory. Inhibitory or excitatory. Otherwise, the only output is from the Purkinje, which is the ganglionic cell layer. So this is what we have as the structure of the cerebellum. Now the cerebellum, we'll visit this shortly. The cerebellum consists of, this is the ganglionic layer, this is the molecular layer. Here you can see this very grandiose type of dendritic tree. This dendritic tree is from the cell body of the Purkinje. Cell body of the Purkinje. So the dendritic tree here form what you call arborizations. Arborizations. You can see continue branching severally, and therefore, as a result of these branches, the Purkinje forms thousands and thousands of synapses. We'll look at that shortly. Thousands and thousands of synapses. I'd like you to look at this cell here. I'll be describing it. We'll be describing also other cells. So what is the structure of the cerebellum? We have the molecular layer. You can see there are very few cells. Very few cells here. A lot of spaces seem to be empty. These spaces are actually the dendrites, consist of dendrites of the Purkinje and also dendrites